So hello everyone, we are back to the online classes. Okay, and today's topic is introduction to autonomic nervous system. Okay, so we have completed general pharmacology, and from today on we will go with autonomic nervous system. So in that series of the autonomic nervous system, the first lecture is introduction part. It will be followed by cholinergic system, and it will be followed by adrenergic or sympathetic system. Now this autonomic nervous system is very important as far as pharmacology is concerned, because many of the drugs that are affecting this autonomic nervous system have a widespread application over many of the major organ systems, including cardiovascular system, central nervous system, endocrine, and many more. So a detailed knowledge of autonomic nervous system and the drugs that affect autonomic nervous system is very important. So we'll start with introduction first. So this is the image shows the organization of the nervous system. Okay, in that you can see that the nervous system is divided into two parts: the central nervous system and the peripheral nervous system. While the peripheral nervous system itself is divided into two parts: autonomic nervous system and somatic nervous system. And this autonomic nervous system is again divided into two main parts, and nowadays the modern textbooks also consider the third part, and that is the enteric nervous system. But the main two divisions of the autonomic nervous system include the sympathetic and the parasympathetic nervous system. So this is our basic organization. This is our basic organization of the nervous system, in that CNS, PNS, ANS, and somatic. Now this somatic nervous system, it supplies or innervates the muscles and skin, which controls our voluntary movements, and the ANS, the involuntary component, which regulates mainly the visceral part of our body. It is divided into two parts, that is sympathetic and parasympathetic. So moving with this, first, the difference between the somatic and the autonomic nervous system. So the somatic nervous system, it innervates mainly the skeletal muscles, which is involved in contraction of skeletal muscles and movement. Then the neurons, these of somatic nervous system, they are mainly located within the CNS. The nerve fibers, they are myelinated type. um, the ganglia are absent. The main neurotransmitter is acetylcholine. And when you cut, whenever there is any injury, or when you cut the nerve that is supplied to the skeletal muscle, there is paralysis and atrophy of the skeletal muscle. In the autonomic type, all other organs other than skeletal muscles, they are supplied by this particular autonomic. It can be parasympathetic or it can be sympathetic. The neurons, they are mainly located within and outside the CNS. The nerve fibers, the preganglionic ones, they are myelinated. Postganglionic ones are non-myelinated. Ganglia are present. Neurotransmitter, depending on the type, it could be either acetylcholine or it could be any is norepinephrine or noradrenaline. Both are one and the same. And when you cut the autonomic connection to the particular organ, usually the organ functions on its own. So there is no change in the activity. So this is the major difference between the somatic and autonomic nervous system. So just like the somatic nervous system, the ANS also consists of an afferent fibers, a nerve center or neuron body, and efferent fibers. So autonomic afferents, the most visceral nerves, they are mixed nerves. That is, they carry both myelinated and non-myelinated fibers, and they carry both afferent as well as efferent fibers in a single trunk. So these cell bodies of the afferent fibers, they are located in the dorsal horn of the spinal cord or in the sensory ganglia. They mediate visceral pain as well as cardiovascular, respiratory, and other visceral reflexes. So basically, the autonomic or involuntary organs which are being controlled by the central nervous system, that organs they send their efferents via this particular autonomic tracts. Okay, and the cell bodies of these neurons, they are located in the dorsal horn of spinal cord or in the sensory ganglia. The central connections or the central neurons. So there are no exclusive areas in the central nervous system which are there for the autonomic nervous system, but widespread locations and widespread neurons are involved in the regulation of the autonomic nervous system. The main organ or the main center which regulates all other autonomic connections, that is hypothalamus. It is the master structure which regulates all the autonomic connections in our body, while the autonomic centers, they are located in the midbrain like Edinger-Westphal nucleus, medulla oblongata, and the lateral columns of the spinal cord. So all these are also autonomic centers. So moving on, autonomic efferents. Um, Motor limb, that is the outflow of the ANS, is anatomically divided into two parts. It could be either sympathetic or it could be parasympathetic. So two parts are there, uh, sympathetic and parasympathetic. Many organs receive both, like majority of the organs, say for example heart, say for example respiratory smooth muscles, the intestinal smooth muscles, the bladder muscles, all these, majority of them, they receive both the sympathetic as well as parasympathetic innervation. So at any given point of time, the level of activity of an innervated organ depends on the sum total of the sympathetic and parasympathetic activity. So this structure shows the anatomical outlay of the somatic, that is this, the motor neuron is there in the CNS. As I told you that the motor neuron is present in the CNS. From there, directly the fibers, the efferent fibers, they directly innervate the skeletal muscle end plate, and the neurotransmitter here is acetylcholine. Okay, for all these, for both the somatic as well as the autonomic nervous system, there has to be a neurotransmitter. So what is neurotransmitter, what is neurotransmission, that we will see in the next few slides. So at this point of time, just remember that in the somatic or in the voluntary nervous system, the neurotransmitter is acetylcholine. Okay, so this is it. Then comes our the autonomic nervous system. Yes. So this autonomic nervous system, it is divided into two parts, the parasympathetic and the sympathetic. Now, in the autonomic nervous system, there is always a efferent neuron which sends a fiber which is known as preganglionic fiber with synapse in the ganglia. This is the autonomic ganglia, and the autonomic ganglia sends postganglionic fibers to the effector organ. The effector organ could be any of these: heart, blood vessels, viscera, eyes, glands, etc. Okay, so same goes for sympathetic. The cell body is there, the preganglionic fiber, ganglia, and the postganglionic fiber. Now if you can see that the preganglionic fibers, they are longer in parasympathetic nervous system while they are shorter in sympathetic nervous system, and the ganglia, they are located close to the effector organ in parasympathetic, while in sympathetic the ganglia are located far off from the effector organ, hence their postganglionic fibers are longer. Okay, in parasympathetic nervous system, the preganglionic parasympathetic neurotransmitter is acetylcholine. So these preganglionic fibers at junction with the ganglia, they release acetylcholine as a neurotransmitter. Also, the postganglionic fibers which arise from the autonomic ganglia, when they synapse with the effector organ, the neurotransmitter released is acetylcholine. So basically, acetylcholine is the one and only neurotransmitter in the whole of parasympathetic nervous system. While in sympathetic nervous systems, all the preganglionic fibers they release acetylcholine with their synapse with with sympathetic ganglia. Okay, it is the difference lies here in the postganglionic fibers. The postganglionic fibers, majority of them, they release noradrenaline. Some postganglionic fibers, like those that innervate the sweat glands, then the smooth muscles of the skin, the blood vessels of the skin, they release acetylcholine. Those postganglionic fibers they release acetylcholine. It is a minor one, but the majority of them they release noradrenaline. While one special circumstance is there, and that is the adrenal medulla. The adrenal medulla, when stimulated by this preganglionic fibers, it releases adrenaline in the bloodstream. Okay, it releases adrenaline in the bloodstream. So this is the this is the anatomical structure of the as well as the organization of the somatic and autonomic nervous system. So I hope this is clear, because you should be able to tell that uh, which neurotransmitter is released by preganglionic as well as postganglionic parasympathetic fibers. Same goes for preganglionic and postganglionic neurotransmitter in sympathetic nervous system. So this should be very clear. If not, just pause the video here and go back little bit and like hear it again, see and understand the structure, so you get clarity about the concept. So moving on, neurohumoral transmission. So just two slides back I told you that we will be talking about neurohumoral transmission in detail. So neurohumoral transmission, what is it? Previously, it was thought that, okay, first the terminology. The term means that nerve transmit their message across the synapse or junction by means of releasing chemical substances or chemical messengers. These chemical messengers, they are known as humoral messengers. Okay, so basically what they mean to say is that the message that is initiated in the central nervous system, it is transmitted across the synapse by means of releasing chemicals. Okay, that is neurohumoral transmission, because both nerves and hormones they are involved. Initially, this was thought to be electrical. So it was believed that the nerve endings uh, change the electrical impulses. Okay, they change the flow of ions and generate some form of current which is transmitted across the junction to the post synaptic effector or neuron, whatever it is. But Otto Louis and his colleagues, Otto Louis and Eliot, they showed that this process involved the use of chemical substances, hence the term neurohumoral transmission. So what are the steps involved in neurohumoral transmission? Five major steps are involved in neurohumoral transmission. Now this goes same. This all these five steps they are involved in both parasympathetic as well as in the sympathetic nervous system. The first one is impulse conduction. Second one is transmitter release. Third one is transmitter action on the post junctional membrane. Fourth one is post junctional activity. And finally, destruction of the transmission or termination of the transmitter activity. So first is impulse conduction. Okay, a resting transmembrane potential in a nerve is minus 70 millivolt with minus being inside, and this is established by a high potassium permeability of the axonal membrane. Now this is basic physiology, so this you must have studied in your physiology. Okay, stimulation or arrival of an impulse causes a sudden increase in sodium influx. So sodium comes inside. So now the inside of the neuronal membrane, it becomes positive, which is known as depolarization. Now these the action potential thus generated sets up local circuit currents which activate adjacent ion channels, and thus the impulse travels. So basically here what we see is that whatever impulse that has been raised by a neuron, okay, transmits itself along the axonal membrane. That is impulse conduction. Once the depolarization reaches the end fibers of the axon, it leads to release of transmitter. Okay, so at the end what happens is that this impulse conduction leads to release of chemicals in the synapse or junction. So transmitter is stored in the synaptic vessels in the prejunctional nerve endings. The arrival of impulse leads to the depolarization. Okay, the membrane depolarization that is set up by sodium influx. What happens is it activates or opens the calcium pumps, okay, the calcium channels. So what happens is the calcium from outside enters uh, the neuronal membrane. It enters the fibers, and this calcium then binds with the synaptic vesicles and facilitates the release of this synaptic vesicles into synapse, a process known as exocytosis, where the membrane of vesicles fuses with the postjunctional membrane and the contents are released into the synapse. So this release process is can be modulated by the transmitter itself as well as various other factors. Now what is this that I will how it modulates, how the transmitter modulates the release of the transmitter from the end fibers, that I'll show you in the last slide. Okay. So third is transmitter action on post junctional membrane. The release transmitter then combines with the specific receptors present on the post-junctional membrane, and depending upon the nature of the transmitter released, it can lead to either EPSP or IPSP. EPSP is excitatory post-synaptic potential, while IPSP is inhibitory post-synaptic potential. Excitatory postsynaptic potential leads to increase in permeability to cations. Cations mean sodium, calcium. Influx causes depolarization followed by potassium efflux. IPSP on the other hand, it increases permeability to anions like chloride, so that the membrane hyperpolarize and an inhibitory postsynaptic potential is generated. The fourth is post-junctional activity. Now, if the excitatory postsynaptic potential is of sufficient magnitude that if it can generate a postjunctional action potential, then it results either, if it is a nerve fiber, then it can lead to nerve impulse conduction. If it is the effector organ is a gland, then it can lead to secretion from the gland. If it is a smooth muscle, then it can lead to contraction of that smooth muscle. So depending upon the effector or the structure present post synaptically, the suprathreshold EPSP can lead to some sort of response, while an IPSP stabilizes the membrane and no activity occurs. And finally, the last part of neurohumoral transmission is termination of the transmitter action. So action of the released transmitter can be terminated by following ways. We need to terminate the transmitter action because we do not want continuous stimulation. We do not want continued action. Once the message arrives, it has been uh, transmitted across the junction, then the messenger needs to go away. So here the transmitter is our messenger, so it needs to go away. So in one of the following ways, we can terminate the action of release transmitter. The first is we can degrade the transmitter enzymatically. Example is acetylcholine. Acetylcholine's action is terminated by the enzyme acetylcholine esterase. It hydrolyzes this acetylcholine, and it is the most efficient way to terminate transmitter activity. Second is reuptake by the specific transporters, like for norepinephrine we have norepinephrine transporter, for serotonin we have serotonin transporter, and for dopamine we have dopamine transporter. And finally, the third is diffusion of the transmitter from the synapse in the adjacent areas, where from where it gets, either it can enter the bloodstream and metabolized by enzyme, or it is picked up by the adjacent neurons or adjacent supportive structures, where again inside the cell it gets hydrolyzed by the agency of enzymes. So the difference between the parasympathetic and sympathetic, we will see. Origin, that is parasympathetic, originates from the craniosacral outflow. So parasympathetic outflow is in the 3, 7, 9, 10 cranial nerves, as well as the S2, S3, S4 fibers of the spinal cord. Distribution. So parasympathetic distribution is mainly related to the head, neck, and the trunk, that is involves thorax and abdomen. But otherwise, it is not present. Ganglia. The As I told you in the figure, that the parasympathetic ganglia are mainly located either on the effector organ or very close to it. The postganglionic fibers are short, and the ratio of preganglionic to postganglionic fibers is about 1 is to 1 or 1 is to 2. The neurotransmitters are, primary neurotransmitter is acetylcholine, co-transmitters are VIP, that is vasoactive intestinal polypeptide, and nitric oxide. Now what is this co-transmitters, that we will see in the next slide. Sympathetic, it is originates from the dorsolumbar outflow, that is T1 to T12 spinal nerves, and L1, L2 and L3, in some cases L4 spinal nerve. Distribution, wide distributed, that is almost all over the body. Ganglia, they are located away from the effector organ, so the postganglionic fibers are long. The ratio of the preganglionic to postganglionic fiber is about 1 is to 20 to 1 is to 100. And the primary neurotransmitter here is noradrenaline, while the co-transmitters are neuropeptide Y and ATP, etc. So these are the differences in between the sympathetic and parasympathetic nervous system. So last part of this introductory lecture is, what is co-transmission? So initially it was believed that one neuron leads to release of one neurotransmitter. So one neuron, one neurotransmitter. That was the concept. But the subsequent studies and detailed studies have now shown that majority of the neurons, they not only release the one predominant transmitter or the main transmitter, but in addition to that, they release many other neurotransmitters as well. Those other neurotransmitters, they are known as co-transmitters, and these co-transmitters, they are also involved in transmission of message across the junction. And this phenomenon of the co-transmitters used as messengers or help in delivering the message, that is known as co-transmission. So in the autonomic nervous system, besides the primary transmitter ACH for parasympathetic and noradrenaline for sympathetic, neurons have been found to release others like purines like ATP, adenosine. Then you have peptides like VIP, NO, neuropeptide Y, nitric oxide, and prostaglandins as co-transmitters. So in most cholinergic neurons, along with ACH, vasoactive intestinal polypeptide is and ATP for sympathetic nervous system is released. The transmitter at some parasympathetic site is NO, nitrinergic nerves. And finally, what are the functions of co-transmitters? So co-transmitters may be involved and serve as same function as the main transmitter with a different time course of action. They also may exert a trophic effect on the synaptic structures. So that is, this is the, okay. So what I have told you that I will explain via this figure. So this is our preganglionic nerve fiber. This is our synapse and this is our post-junctional. In this case it is a nerve fiber, but it could be a post-junctional effector organ as well. So whenever there is an impulse, so this is our impulse comes. So first step, impulse conduction. Second step, release of transmitter. Third step, binding of this transmitter to the post-junctional membrane. Fourth step, either EPSP or IPSP, that is post-junctional activity. And the fifth step is termination of transmitter action. So here this orange color is our primary transmitter, while green is a co-transmitter. Okay. Now how transmitter modulates their own release. So you can see that the transmitter, this or this, they can bind to their receptors located prejunctionally, okay, or presynaptically. So these presynaptic receptors, they usually decrease or increase the release of transmitter, or their own release, that can be modified by this pre can be modified by this presynaptic receptors. So this is all about the neurohumoral transmission, the the concepts of sympathetic and parasympathetic nervous system, the major transmitters that are involved in the autonomic nervous system. So that is all. Thank you.